Noushka Reiter. I work here at the Royal Botanic Gardens Victoria at the Australian Gardens Cranbourne site, and I manage the orchid conservation program here. Many of Australia's orchids are threatened and endangered because they are simply reliant upon so many other factors in the environment in order to flourish and grow. They're reliant upon a specific relationship, often with a pollinator, in order to set seed. They require mycorrhizal fungi in order for the seed to germinate and grow. And unfortunately, due to historic pioneer farming, we've lost many of our native habitats. There are about 1,700 species of orchids here in Australia, of which about 17% are federally listed. So without our help, they won't really be here for the future. So let me introduce you to some of the characters that we have in our shade house at the moment. So this is the gorgeous Pterostylis tenuissima, known really only from swamps. Mycorrhizal fungi actually grows in the lateral roots that come out in the bottom of the orchid. And as we're out there in the middle of the swamp, isolating the fungi, just being covered, dripping with leeches, it's a wonderful experience working with this one. And if you come over here, this is Caladinia concolor. We've found that it's pollinated by a wasp. And unlike most of our wasp-pollinated Caladinias, which are generally sexually deceptive, so the Caladinia emits pheromones, which smell like a female, and they attract the wasp, and it comes in and mates with the orchid, and in doing so, it pollinates it. This one is quite different. So if you smell it, it's this lovely mandarin flavour, and we now have the wasp coming in, and it's actually attempting to try and feed off this orchid, so we think it's food deceptive. I'm really excited to be showing you this species. This is Caladinia audacei, the order spider orchid. It's down to only 5 in the wild, and this is the first time it's flowered in cultivation. And we're incredibly excited to be able to be doing some hand pollinations between these and a couple of the remaining wild plants this year in order to get much needed seed for propagation. What I'm doing here is hand pollinating Caladinia concolor, and the pollen is just in behind these little anther caps here. On orchids, and particularly in Caladinias, the stigma sits just behind the anthers here on the column, and the stigma, of course, is the girly parts. And so if I just put the pollen on the stigma there, we've hand pollinated that little guy. And that'll now form a lovely pod and set seed, and we'll collect that in about 6 weeks. Once seed's been collected in the field, it's into the lab. The seed is then dried here and stored in the fridge. Next, the mycorrhizal fungi is isolated and cultured here in the lab. We take the seed and we bleach it. We then rinse it in sterile water. We then add on our mycorrhizal fungi and put them aside to germinate. So next we take our seedlings when they're about 3 months old, and we flask them into a layer of vermiculite to grow in, and oatmeal agar and yeast for food. Next, the orchids are taken here into our nursery where we plant them out, and we grow them to a suitable size, like these guys here. And then we get the absolute joy of reintroducing these plants into the wild in suitable habitat where the pollinator is present, and hopefully we get to save these species from extinction. All sorts of wonderful people volunteer, largely from the Australasian Native Orchid Society. We have lovely retired scientists that volunteer here in the lab, and we have a team of people that come out every year helping us both survey for new orchid populations and reintroduce orchids back into the wild. We've propagated about 30 species of threatened orchids. Of those, we've reintroduced about 20 species so far, and about 16 of those are doing well. I think that people, do care about the environment and that we can make a difference. And I think anyone working with threatened species needs to be an optimist. Most of the problems that we're dealing with now we've caused ourselves, so we should be the ones to be able to change that as well.